Since I don't know you very well at this point, I wasn't sure just where to aim my presentation. And I apologize in advance if it's too basic for most of you. I'm sure that it's not going to be too complex for any of you. What I will present this morning is a brief discussion of general issues as to why glucose and insulin are potentially important in the context of aging and calorie restriction. In that context, there will be some discussion of the basics of carbohydrate metabolism, which may be very familiar to all of you, and mention of some of the deleterious consequences of hyperglycemia and hyperinsulinism. Hyperinsulinism, as distinguished from just hyperinsulinemia, which is a transient elevation in insulin levels. Hyperinsulinism, on the other hand, is a chronic elevation of circulating insulin. Then I'll describe our findings in our project on dietary restriction and aging in rhesus monkeys, with a brief overview of the design and methods, and then some of the results pertaining to insulin sensitivity and glucose tolerance. And then I'll just wrap up with one or two slides summarizing the data. Now, the very basic physiology of glucose homeostasis begins with the ingestion of a meal and the digestion and absorption of foodstuffs from the gut, which results in an increase in blood glucose levels, which then circulate throughout the body in the blood, and the glucose is then taken up to be utilized as a fuel by all the tissues in the body. And for some of the important tissues, such as muscle and adipose tissue and liver, glucose uptake is greatly facilitated by the presence of insulin. For another very important organ, the brain, insulin is not required, and the brain uses quite a bit of glucose as its primary fuel, and really accounts for a large portion of glucose disposal under normal circumstances. The alternative fuel for the brain is ketones. Now, the insulin is secreted by the pancreas in response primarily to an increment in plasma glucose. Now, the pancreas is a relatively small organ adjacent to the duodenum, and in spite of its relatively small size, it is critically important. And it's important in the context of this morning's discussion because of the presence of pancreatic islets, which contain the cells which secrete insulin. The bulk of the pancreas consists of cells and tissues that are related to digestive processes. But the islets of Langerhans, which represent only about 2% of the mass of the pancreas, have this endocrine function with 4 primary cell types present in the islets. The beta cells being the principal cell type, and they secrete insulin. And they represent about 75% of the mass of the pancreas. The alpha cells secrete glucagon, and that's the other prominent cell type. And then smaller numbers of delta cells and F cells or PP cells secrete pancreatic polypeptide. Insulin is a peptide hormone, and in the process of its synthesis and release, it is packaged in vesicles inside the beta cells of the pancreas, initially in the form of this large molecule. Before it is secreted, this portion of the molecule is cleaved to create insulin, which is this double-stranded portion, and the cleaved portion, which is known as C-peptide or connecting peptide. The insulin molecule itself consists of these two chains, one consisting of 21 amino acids, the other of 30 amino acids. The two chains are linked by disulfide linkages on cysteine residues in the polypeptide chain. The C-peptide represents the remaining portion of the proinsulin molecule, and it's a very useful tool. It has no physiological function that's been identified, but it is released from the beta cells in an equimolar amount to the insulin that's secreted. When insulin is secreted, it enters the portal circulation and goes directly to the liver, where about 50% of it is immediately destroyed. C-peptide, on the other hand, is relatively immune to the catabolic processes of the liver and continues to circulate until it's secreted or excreted in the urine. So when one wants to address the question of how much insulin is being secreted, it's very difficult to tell that by simply measuring insulin levels in the blood, because perhaps 50% has already been eliminated by the liver. But if you measure C-peptide, it's a good surrogate marker for the level of insulin secretion by the beta cell. I'm saying all of this because it will become a little more important for interpretation of some of the data that I'll show in a minute. So, after ingestion of a meal, blood glucose goes up, stimulates the pancreas to secrete insulin, which promotes the uptake of glucose and other fuel substrates by fat, muscle, liver, and other insulin-sensitive tissues. And there's a restoration, eventually, of basal blood glucose levels. In contrast, after a prolonged fast, blood glucose tends to be relatively low. There's minimal insulin secretion under those circumstances. On the other hand, glucagon is released by the alpha cells, which promotes the release of stored glucose in the liver to increase blood glucose levels. So it's a very finely tuned mechanism under normal circumstances to maintain a very well-controlled level of basal glucose. I think I'll skip this one and illustrate it here. Again, insulin's major biological effects are to promote the uptake of glucose by muscle and adipose tissue cells primarily. It also promotes the uptake of amino acids into muscle cells and promotes the uptake of free fatty acids from the blood into adipose tissue cells. It inhibits the formation of ketone bodies by the liver and promotes the formation of glycogen, the storage form of glucose, in the liver. Now, the way insulin works is very simple, and you can see it all on this slide. Greg Carty will explain this in more detail in his presentation tomorrow. Now, all too often in our culture, things go wrong and an individual's blood glucose becomes chronically elevated. And this has myriad deleterious effects for the individual. These include increased glycosylation of proteins and nucleic acids, formation of advanced glycosylation end products. These events are associated with the development of atherosclerosis, with stiffness. The glycosylation of the protein seems to interfere with the function of elastin and other molecules in the connective tissue. It's also associated with retinopathy and a variety of other things that you don't want to have. All too often, and frequently in association with the development of obesity, there's a resistance to the physiologic effects of insulin in the periphery, which stimulates the pancreas to secrete more insulin to overcome that resistance, resulting in chronic hyperinsulinemia or hyperinsulinism. One of the major deleterious consequences of this is an increased risk for the development of diabetes mellitus. In simplistic terms, the increased demand on the beta cells ultimately results in exhaustion of those cells and an inability to keep up with the demand, resulting in diabetes. And hyperinsulinism is strongly associated with abnormal and pathologic blood lipid profiles, the development of atherosclerosis. It's thought to perhaps be a causal link in the development of hypertension, and there's increasing evidence to suggest that hyperinsulinism may be a contributing factor to the development of several cancers. So these observations should give us pause and wonder about how an intervention could favorably affect the regulation of blood glucose levels and the regulation of insulin levels in a manner that would increase our health span and perhaps our ultimate lifespans. And in order to approach some of these issues in an experimental way, it's often prudent to first go to an animal model. And rhesus monkeys, such as the fellow illustrated on this slide, I would say are outstanding animal models for work in this area. I'm a primate and proud of it, and I think that we can learn a lot from studying some of our close relatives. Now, I say close loosely. Rhesus monkeys are in the category of old world monkeys. They're derived from Asia and Africa. And it's thought that they diverged from the hominid line about 12 million years ago. So there's considerable distance, but still great genetic similarity and many physiologic and anatomic similarities to our own species, making them, in many ways, a very appropriate animal model.
Go back one second.
Sure.
That's a good slide.
The great apes, the orangutans, the chimps, and the gorillas are the most closely related to us in a phylogenetic way. The lesser apes, including gibbons and siamangs, are intermediate between the great apes and the old world monkeys in terms of relatedness. The new world monkey line, that is those primates living in Central and South America, diverged somewhat longer ago, and then the prosimians, such as the lemurs and the lorises, are even more distantly related. The rhesus monkey